Hello, class, and now that you all know about mitosis and meiosis, remember mitosis we get is just the regular cell reproduction cycle. Meiosis is a little bit different. In mitosis, we got identical daughter cells, two identical daughter cells. In meiosis, we get four unique individual daughter cells. We're going to now look at how that impacts what we see. What is the relationship between meiosis and genetic variation? Here's a picture of a family, for example, here. There's a very, very, very large family. Here's the mom and the dad, and here's lots and lots of kids. None of the two of them look just alike. They're all unique individuals. Can't quite imagine having that many unique individuals in one family, but this is a large family. There's one mother and one father, the biological parents of all these kids, and all of them are unique and different. So how does that happen? Why are brothers and sisters different from one another? What is the relationship between meiosis and genetic variation? So we're going to talk about that today. Let's do a quick review about meiosis. Remember, what are the four gametes that could be produced from a parent who is heterozygous for two different traits? Heterozygous for this A trait and heterozygous for this B trait. Remember, just a little review of foil. We can pick out those four different gametes. We can do the first two. We can do the outside. We can do the inside. And we can do the last. Those are the four different gametes, the four different ways that we could combine these two different traits and have different combinations. So how does this relate to mitosis? So let's look at these steps in meiosis. We start with prophase 1, the very first step. Remember, our cells are diploid, okay? So we have the full 2n numbers of chromosomes. They're not haploid like the gametes, okay? So we start with our diploid cell. Here's an example. We're going to use in this example 4 chromosomes. If we were looking at a human cell, we would be looking at 23 chromosome pairs or a total of 46 chromosomes. We're going to reduce the number to simply 4, and we'll end up with 2 pair because that's just a little bit easier to work with. Remember that each chromosome consists of 2 chromatids each. And these chromosomes in prophase 1 are going to form tetrads. And how do they do that? Well, look at this one here and this one here. Those chromosomes are what we call homologous chromosomes. Okay? Homologous chromosomes. And we call them homologous because they are the same length. And they are going to, this chromosome, they're going to find each other and they're going to form a tetrad together. Likewise, we're going to have these 2 chromosomes are going to find each other. They're going to pair up to form a tetrad together because they are also homologous. These are similar in length. So here we have our tetrads. Here's a tetrad because the homologous pairs of chromosomes formed these tetrads. So in metaphase 1, the tetrads line up along the equator. This is different from mitosis. In mitosis, there are simply chromosomes lining up along the equator. In meiosis, metaphase 1 sees tetrads lining up along the equator, paired chromosomes. Now, one thing, too, to remember, these formed a tetrad in this fashion. I have the big A over on this side, I have the little a's on the right. It could have very well been flipped. Either way, it could happen either way. So that is something we'll talk about a little bit later. Also, if you'll remember, the spindle fibers are attaching to the centromeres here of each of the chromosomes within the tetrads. And then those are held together at the centrioles at the ends. And that's getting these tetrads prepared for the next phase, anaphase. Here we have anaphase, our very next phase. We have the tetrads are separating. This is different from mitosis, where the sister chromatids separate. In meiosis, anaphase 1, the tetrads separate. So we have a tetrad going this way and a tetrad going this way for the A's, and the B's likewise. They could have split differently. We could have had the little a over here and the big A over here. Again, we'll talk about that a little bit later. In telophase 1 of meiosis, we now have our cleavage furrow happening for the animal cells, right? We have the formation, the near formation of two new cells, daughter cells, and kind of temporary because we're going to go on to the second part of the meiosis pretty soon. These are both haploid cells, and these are the different gametes that can be forming, big A and big B, or little a and little b. And remember, we had said before, there should be 4 possibilities, right? So where are the other 2 possibilities coming from? We're going to talk about that next. Remember, I said earlier that you could have these switched, right? The little a doesn't have to go to the right and the big A to the left. We could have these actually going different directions. So let's look at those other options. So here we have version number 2. What if we had these chromosomes lining up so that the little a went this way and the big A went this way? One thing important to remember in all of this, this is independent assortment, sort of the example of it. We could have the little a going this direction and the big A going this direction. That would be independent of what the B's do. We could have big B going this direction and little b this direction. They are separate from each other, so they are assorting independently of each other, but it is pure chance whether little a will go this way or that way, or big A this way or that way. And that is where we get our 4 different possibilities. The tetrads could have separated in a different order, and it is totally by chance, a 50-50 chance for each one of those traits. This would be what would have happened had we had these assort differently, where we have the little a on this side. We would get these other 2 different gametes. We would get the little a big B and the big A little b, just depending on the direction and the way that each of these individual tetrads form together, and the chance that the spinofibers were shortening which direction for which one, which way they were getting pulled. That would just be a 50-50% chance that they could go either way. So just a quick review. We have our the idea of segregation. Remember, segregation, meaning that we get one allele from the mother and one allele from the father. And in this case, here's where they're coming from. One from the mother and one from the father for that trait. One from the mother, one from the father, or vice versa from that trait. That's the segregation part. Remember, we talked also about independent assortment. And that is where this trait, the genes are going to assort independently of this trait. They are not dependent on each other. Whether these go which direction doesn't affect which direction these are going to go. Now let's look at the sex-linked genes. So when we talk about the human chromosomes, we have 23 pairs total, okay? 22 of those pairs are autosomes. Autosomes, you'll see right here, if these would be autosomes, they are the ones that are not the sex chromosomes. And those are, you have 22 pairs of autosomes. 1 pair are the sex chromosomes. So here's the pair that would be the sex chromosomes. You have one from your mother, one from your father, and if you'll remember, let's make this dinky little, there's the little y, if we would call that our very small little y. The father would either contribute, in this case, here he's contributing the X, or the father could contribute this Y. Those are then the sex-linked genes. Remember, if the father contributes a Y, the offspring will be a boy. Okay, and if he contributes the X, it would be a girl. And remember also the Y carries very little, very, very little genetic information, so it is not able to counteract any genetic information that the mother would pass along with the X chromosome. Now let's continue on with the process meiosis, we're going to now look at meiosis II. And remember, meiosis II, all the different steps in that is very much like mitosis. We are going to start with these two cells, and remember, we'll have prophase, we'll have metaphase, where these chromosomes will line up along the equator, and then we will have anaphase, where the sister chromatids pull apart. We'll have one pulling this way, one pulling this way, one pulling this way, one pulling this way, and same with these, one this way and one this way, one this way and one this way. If we sort of look at that, think about it like dividing, and we would end up with 1, 2, 3, 4 different cells total, 4 unique daughter cells in the end of this meiosis. Now remember, we're getting these 4 different gametes that are being produced by an individual's cell, diploid cell, because we have those other possibilities. Remember the sort of option number 2, these could assort in different directions. They're sorting independently, and if they assort in the other way, we have big A over here and little a over here, we're getting those other potential gametes. So we end up with 4 potential gametes that a sperm could produce, and 4 potential gametes, or not that the sperm could produce, but that could be produced for a sperm, and 4 different gametes, gamete combinations for an egg. That is looking at 2 pairs of chromosomes. What if we were looking at 3 pairs of chromosomes? We get 8 different possibilities if that's the case. So remember, human beings have 23 pairs of chromosomes. And if we take that rule 2 to the factor of how many chromosomes you're looking at, that will give you the different possible combinations that could be seen in an egg, or those different possible combinations that could be seen in a sperm that the parent produces. 2 to the 23rd, when we look at that number, it is 8,388,608 different possibilities for a single egg or for a single sperm. That's a lot of different possibilities. So now, let's look at what happens when we combine an egg with a sperm. We have 2 to the 23rd possible eggs, 8 million plus different possibilities there, with 8 million plus different possibilities for the sperm. And when we combine those together, there are over this many possible offspring that could be produced from one mom and one dad. Here's hundreds, thousands, millions, billions, 70 trillion different possibilities. That's a lot. Wow. You're so lucky that you were born. You are very unique and very special. Only 1 in 70 trillion. And this is based just on independent assortment alone. The fact that each of the chromosomes assort independently give us this many different possibilities and this many different possible combinations. So here's some key ideas so far to take away. One, when we're looking at Punnett squares, we're really looking at chromosomes, okay? When we think about the way that these chromosomes are combining, I want you to remember the different key events in meiosis, especially meiosis 1. That's where the big differences are between the mitosis and the meiosis is in meiosis 1. We have in the metaphase 1 of meiosis, we have tetrads forming. That doesn't happen in mitosis, but in meiosis we have those tetrads that the homologous chromosomes form those tetrads together, and that's where we get independent assortment. When they form together and they form independently of each other. The different traits are not dependent on each other. Then when we look at anaphase 1, where the tetrads are then pulling apart, they are segregating. You're getting one parent, you're going to end up having one parent giving one allele to the offspring, and the other parent will be able to be contributing another allele because of that anaphase. Also, the very last thing to remember is that the number of different assortments is just 2 to the n, where n equals the number of chromosomes. So in a fruit fly, it'd be 2 to the 8th power. That would be how many different assortments that could happen for the gametes of that individual. For human beings, it happens to be 2 to the 23. Now we're going to take a quick look at sexual reproduction and variation. What's the big deal about sexual reproduction anyway? Sexual reproduction is just the process whereby haploid gametes from two different individuals come together. That's fertilization. The haploid gametes are the sperm and the egg. When they come together and fertilization happens, they create a new unique diploid individual. This process actually presents many challenges, not the least of which is finding a partner in the first place. Other challenges to this process: what if the partner you meet isn't just like you? Or what if the partner you meet would rather eat you instead? More challenges to this process of sexual reproduction. What if your partner actually does eat you, such as is in the case with praying mantids? What if you finally meet a partner of your same species, of the opposite sex, and they're not exactly mating material? More challenges to sexual reproduction. Sometimes you and your partner just move too slowly or live too far apart to actually find each other. That's a big challenge. What if your partner is not in the mood, even though you do your very best to look attractive? And even if you do find an appropriate partner, sometimes those babies just don't turn out the way you'd expect. What about asexual reproduction instead? Asexual reproduction occurs in every single kingdom. It's very, very common in archaea and eubacteria. Protists, an amoeba, is an example of an organism in the protist kingdom that reproduces asexually. Yeast and mushrooms carry on asexual reproduction in the fungi kingdom. Strawberries, potatoes, aspen are examples in the plant kingdom. Sponges, planaria, and starfish are examples in the animalia kingdom. Here is what happens for asexual reproduction. Basically, cell division. You get two identical daughter cells. So if it's this simple, why is it that sexual reproduction occurs? What is the big deal about it? Here is the big deal: variation. Variation is the key to sexual reproduction. Sexual reproduction results in offspring that are not identical to either parent. That happens to be the greatest strength and the greatest weakness of sexual reproduction. Because of natural selection and a changing world, we have a changing environment. Variation is precisely what has allowed life to thrive in nearly every corner of the planet. That variation, in combination with the natural selection that occurs, allows life and thriving. How does sexual reproduction provide for variation? This is a key point to remember, several key points to remember. The combination of the two different gametes, that provides for variation, right? We have those two different gametes that can combine together. We also have independent assortment in meiosis that gives us a lot of different variation. We also have something, we've been talking about these things, but here is another really key point to remember. In addition to all of the different possibilities that we get from the fact that two gametes are combining. Each of those gametes, because of independent assortment, is given about 8 million plus different possibilities of their own combination, just because of independent assortment of the 23 different chromosomes in humans, for example. We also have this crossing over that adds even more possibilities to the combination, okay? What is crossing over? In meiosis, when we have tetrads that have been formed, here are the tetrads, here's an example of a tetrad. During that formation of the tetrad, a little piece of one of the genes crosses over another one, and they switch genetic material, so that you end up getting, when the tetrads pull apart, this is carrying a little piece of the genetic information from this, and this chromosome is carrying a little piece of genetic information of this. It's like a little shuffling of genetic information when they're in the tetrads, so that when you get your gametes, you have way more than just 2 to the 23rd gametes that are possible. It is many, 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 many times more than that. So not only do we have this great variation possibility because of the 2 different gametes and because of the independent assortment, but we also have this factor called crossing over that gives us even more different possible combinations. Here are some more ways sexual reproduction provides for variation. Errors in meiosis. Meiosis is not a perfect process. It does not always proceed without error. A couple of different examples of that. Non-disjunction, when the chromosomes don't actually separate. Polyploidy. Polyploidy is where we get chromosomes that are a different number than just the 2N. Sometimes we get monosomy, like just a 1N, or a triploid, or quadruploid, where we have 3N or 4N numbers of chromosomes. That's much more common in plants. Here, bananas and watermelons, seedless watermelons, are examples, or other seedless fruit as well, are examples of a monosomy or trisomy genetic combination, where you're getting simply 1N or a 3N possibility in the numbers of chromosomes. We also have things that we will talk about in later sections, chromosomal mutations and DNA level mutations. Those also provide for variation. So in conclusion, asexual reproduction does have advantages, especially when the environment is extremely stable over time, which is rare. That almost never happens. So asexual reproduction in a really stable environment will be advantageous. However, sexual reproduction and variability, the variability it produces, is ultimately an advantage when the environment is unstable over time. Therefore, it ultimately is an advantage because the environment is unstable over time. We find that even organisms that can produce asexually and sexually, they prefer the sexual reproduction. This is always preferred because we have a changing and unstable environment. Even though some specific variations are beneficial and some are harmful, not all mutations are beneficial, certainly, or some variations are not advantageous. The variation itself is so advantageous that it ends up being built into the system at many levels, and it is what is chosen and selected we see in nature all the time. So even due to the difficulties and hardships of sexual reproduction is preferred because of the variation. Advanced ideas, research organisms that have unusual reproductive patterns or can switch their reproductive patterns. Explain when, why, how does this happen? There are all kinds of really fascinating and unusual sexual reproduction patterns that you can find in life on this planet. It's really amazing, really interesting, really fascinating, and I look forward to seeing what kinds of information you come up with.